My name is Tom Hanson. I'm the specialty chief editor for Microbial Physiology and Metabolism in Frontiers in Microbiology. So my current research deals with phototrophic bacteria, which most people think about as pond scum. And what I'm interested in is how they actually extract energy from their surroundings. So broadly, it's microbial energy metabolism, and microbial physiology is how microbes make a living in the world. Microbes for most people typically mean things that you can't see, which for human eyes is anything that's smaller than 0.1 millimeter apart. But there's lots of examples now of microbiology that we can see from space. So I work on sulfide oxidation, and in fact you can see sulfide oxidation occurring in certain areas of the ocean from space by satellite imagery by virtue of the elemental sulfur that's formed. So what I'm really excited about working on in my lab is we're trying to understand how these organisms actually interact with elemental sulfur. So elemental sulfur is insoluble. It's not in solution. And organisms accessing solid things it's been studied for metal oxides, but it's not really well known for other materials. And we think we have an idea of proteins that are now involved in this process and that might be involved in both forming and consuming this insoluble material. So it's really understanding very basic aspects about how microbes interact with solid materials. So my research is actually very basic, and it's not necessarily that in the next year there's going to be a product or that there's going to be an application. However, these types of organisms that interact with solid surfaces are employed in biomining. They're employed in bioremediation. So we want to actually convert toxic compounds like hydrogen sulfide to non-toxic compounds like elemental sulfur. So the more we understand about that process, the more efficient we might be able to make it. My dream would be that I could just fund my lab for the next 20 years and do what I want to do. But no, seriously, I would like to, I don't think about application every day, but I would like what I do to find application somewhere. And thinking about, again, this idea of microbes interacting with surfaces, this is important in pathogenesis. And it turns out that some of the proteins that we found actually are very distantly related to proteins that appear to interact with mammalian cells. So there may be a universal aspect to microbial interactions with surfaces that we wouldn't have understood before if we hadn't looked at sulfur oxidizing microbes. I am, full admission, I am a second generation microbiologist. So my dad is a microbiologist, although what I started out doing was working on methane oxidation. That was what he worked on in his lab, and so I kind of grew up in the family business. And then when I went to graduate school, I wanted to distance myself. So I went and I started working on phototrophic microbes because they weren't E. coli, which had been studied for 50, 75 years. I didn't think there was all that much new to be learned there, and I wanted to work on systems that are a little bit more out at the fringes and maybe more environmentally relevant. And then in my postdoc, that's where I started working on the organisms that I work on today, and I got into sulfur metabolism as the result of an experiment. We knocked out a gene that we thought might be involved in carbon metabolism. The phenotype was sulfur metabolism. And then 15 years later, that's where I am. Yeah, so the types of organisms that I work on, sulfur metabolism for me means taking reduced sulfur compounds, like hydrogen sulfide, everybody knows that as rotten egg smell, right? And converting that to elemental sulfur, which if you've been to or seen pictures of Yellowstone National Park, that's where Yellowstone gets its name, is elemental sulfur and the rocks and formations around there. You think about volcanoes, all that yellow material, that's elemental sulfur. So what I'm really also trying to understand is, in some ways, how these planetary elemental cycles work and what the role of biology is in creating those. Sulfur is one example. Another big one is oxygen. If you take a breath in and then you exhale that, the question I always ask people is, how much of that oxygen that you just took in was actually made by microbes? And the answer is about 50%, because 50% of the oxygen production in the world occurs in the oceans and is catalyzed by microbial organisms. Right. So how does my work, or how does the work of sort of microbial physiology and biogeochemistry really integrate into sustainability, right? So there's a lot of ideas out there about either geoengineering or things to affect climate change. And a lot of it is based on, oh, there's a microbe out there that does this, like a microbe that eats methane. The problem is that we could almost never grow enough of those microbes to actually make any sort of dent in the methane budget of the atmosphere, right? So one is to inject a little bit of reality in that. And two is to actually understand and constrain what natural populations are doing. How much do they contribute to sulfide oxidation, to CO2 drawdown, or CO2 efflux? I've done some work in microbial tundra looking at microbes that break down soil organic matter. And as temperature rises, it's actually expected that soil organic matter in tundra is going to become a major atmospheric CO2 and methane source. So understanding the role of microbes in that process is going to help us to better constrain and make predictions about what's going to happen. In some ways, so examples of microbes that might, be beneficial in terms of sustainability. One thing that's actually been worked on a lot are biofuels, right? And so one thing that's being looked at is engineering microbes, and that is engineering their physiology, their biochemistry, which is what I'm interested in, to make value-added products and to make them from CO2 that's directly in the atmosphere rather than from petrochemical feedstocks. That's the one that I think has the most potential in the short term to actually make an influence and make these things now from new carbon rather than old carbon that's been in the ground for hundreds of millions of years. No, in these types of systems where we're talking about biofuels, that is, we would set them up as reactors, which food companies and companies, chemical companies have already been doing to make sort of value-added products. In that case, we would set up a reactor with the goal of making, say, ethanol from steel plant waste. And in fact, there are plants that are already doing this. There's a company that's out of New Zealand called LanzaTech, which has full-scale ethanol production at several steel factories in China that are using the CO2 emissions from that to actually fuel ethanol production. So that's a really good question. Why are we not doing more with these microbial processes that could affect sustainability? Some of it is economics. These processes need to be economically viable, right? And in biofuels, in particular, where you're getting that feedstock matters, because a lot of feedstocks like corn, then you're competing for agricultural land for the feedstock that you put into the microbial fermentation to make whatever biofuel you want. We want to avoid those types of competitions, but that's kind of hard to do and avoid those types of trade-offs. So I think economics and also, again, looking carefully at the sustainability aspect. Corn ethanol is another good example of that. So corn for ethanol or biofuels is essentially only carbon neutral if the plant is sitting right in the cornfield, because once you factor in transportation costs for the biomass to get it to the plant and the CO2 that's released making the fertilizer for corn, which typically takes a lot of fertilizer, then it's nowhere near carbon neutral. And that's what most people don't understand about some of these things. So I think that's where some communication and open science is really critical to make sure that that message gets out about what's truly green, what's truly sustainable, what can we do, and what might actually be feasible. So I think one of the things is there's an old adage, which unfortunately was co-opted by the CIA, which is you shall know the truth and the truth shall set you free, right? Data, open information, open protocols are all going to be key to getting solutions and getting science out to industries, out to governments, and out to people that might actually be able to translate it into something. Myself, a bench scientist, I'm probably not going to have the right skill set to take a discovery that I've made and say, oh yeah, we can turn that into microbially produced plastics at scale and make it economically feasible. But if my data is out there in an open and accessible format, it can actually go out and be used. And I think ultimately, even for the most basic scientists like myself that are working on problems because fundamentally we're just interested in them, we'd ultimately like to see those types of things be used, even if it's not right in the front of our brain every day. I've actually thought a little bit about this in terms of whether or not there should be a requirement for scientists to sort of work on projects that have some easily definable social benefit. I don't think so, because we don't know what we're going to discover. And in fact, if you look at penicillin, penicillin was discovered by accident. It was a contaminant that fell into Fleming's dish that started killing off his experiments. And it wasn't Fleming that ended up commercializing that. It actually, in large part, was the federal government and the USDA and fermentation facilities in Peoria, Illinois, that ended up scaling up production to where penicillin could be useful. And I don't think anybody would argue that antibiotics have been, maybe now they're beginning to be a little bit of a problem, but antibiotics at the outset saved millions of people's lives, all because of an accident, a contaminant in a petri dish. So I think that having some sort of undirected open science that leads to serendipitous discoveries, I think that's a good thing. I don't think we all need to be working on applied science. The other way I've thought about this is in a course that I teach, or that in a course that I participate in. One of the things that I have students do is argue on both sides of an issue of what I call an innovation core. In that if your graduate work or was funded by federal dollars, should you then be required to do what I would call a service postdoc, where you work on a problem that's been identified by the national academies as being of national interest? And it's really interesting to see the students' perspectives on this. I'd say that by and large, it's probably about 80 to 90% feel that they don't want that type of restriction and they shouldn't be held to that, even though the money is coming from the federal government that supports their research in the first place. It's really interesting. Maybe that's just American ideas of freedom, but I don't know. So I joined Frontiers because Martin Klotz, the field chief editor of Microbiology, asked me to join. I mean, that's the first thing. It was fairly flattering at that point. But also, the thing that I like is the idea of open review and trying to collaboratively improve science, right? Science doesn't do anybody any good if you publish poorly phrased, poorly executed results, and then nobody's able to actually take that, reproduce it, and do something with it. So I look at my role as trying to help facilitate that improvement. And there, the open and collaborative review process is actually, I think, what attracted me the most. It turns some people off. Sometimes I have a hard time recruiting reviewers to work on papers because it's more effort than a traditional review, but I think ultimately it actually does improve the science. And that's really my target for the journal, is to actually improve the quality of publications in microbiology that are coming out.